Yes,
Okay, perfect.
I'm putting you in the center of the screen.
All right. Well, thank you very much. Thank you very much, and good morning, good afternoon to everyone, depending where you are. And thank you, Paulo, for this opportunity of presenting today. I'm really excited about having this chance to discuss our technology, our company, and some applications of our products. First of all, I will briefly introduce myself. I'm originally from Argentina, based in Spain. I'm an agronomist engineer, and I work in the European team with Biome Makers as a sales manager. I will share with you all a presentation I have here. Please, Paulo, let me know if everything works okay.
Yes, now we see your presentation.
Awesome, great. Well, first of all, the agenda. These are the topics we will be discussing today. First of all, I will introduce Biome Makers a little bit. Then we will talk about BeCrop, which is one of our main tools for analyzing the microbiome. And afterwards, we will jump into the Kairos vineyard case, which is one of the uses of our technology. And then we will discuss some other possibilities that BeCrop can offer to our customers for going above and beyond in terms of soil analysis. Well, first of all, Biome Makers. We are an agtech startup headquartered in Silicon Valley. We started our operations about 6 years ago, and today we have global presence with 2 laboratories, one in West Sacramento and a second laboratory in Europe, in Valladolid, in Spain. Basically, what we do is we develop cutting-edge technology for soils. We have a qualified rockstar team, and of course our vision and mission is to recover soil health worldwide. These are some of our colleagues. Today we have more than 40 colleagues. More than 45% of our teams are PhDs. So we invest a lot in science and R&D and development. So we are proud of them. And of course, as I said, our vision is to recover soil health worldwide, to help both farmers and ag input manufacturers to do this, with the mission of becoming the leading soil check platform. We want to offer technology for modeling this soil functionality so that we can enhance the productivity in all the arable soils across the world. Now, the question is, why the soil? Because this is something relevant for this presentation. The main answer is because our soil is alive. Probably many of you already know this, but there are billions of microorganisms out there in our soils, and those microbes are ready to interact with both our crops and the products we apply to those crops, and to boost the productivity and the sustainability of those crops. Actually, depending on the references we read, we have from 3 to 5 tons of microbes in 1 hectare of soils. Of course, we have different kind of microbes. We have beneficial microbes, pathogen microbes, and commensals microbes. The last category, the commensals, are these microbes that don't have a direct interaction with the plant, with the crop, but they are still important for the function of the community and the ecosystem. And this is quite relevant. For us, microbes are also an accurate marker of tons of processes. I mean, we can analyze microbes as they're based on the function they have on the soil. For example, a mycorrhiza, azospirillum, or any kind of beneficial microbe. But we can also use the presence of a microbe in a community as a marker or some specific process. The fact that the microbe is there alone or together with some other microbes can tell us or can provide us a lot of information about what's going on in the soil. So we believe that we must take into consideration these microbes, and we have to do it in a sustainable and productive way, because these two words are really important today, because we need to be productive, but we must be sustainable. So this is the key. But how can we make the most out of these microbes if we cannot measure them? This is a key question, because as this quote from Peter Drucker says, we cannot manage what we cannot measure. So it's really important to be able to measure and to understand what's going on in the soil in order to be able to take profit or to make the most of that situation. So the solution we come up with as a company in Biome Makers is decoding all the microbial interactions that are happening or occurring in the soil, so that we can unveil a new dimension in soil analysis. It is like if we can open a new window and take a look into that black box that the soil used to be, and see what's going on there. So the result of this approach and the result of the technology that our science department developed is BeCrop. This is one of our products. This is one of our tools. And the way we add value, because I think this is important to understand the results we will discuss later on in this presentation, we can add value by understanding or detecting all of these microbial interactions. And everything starts with a soil sample. For us, a soil sample means a lot of information, because we can extract all the DNA present in a sample, convert that DNA into data, and process that data through a machine learning and artificial intelligence system to come up with a very easy to read yet insightful report that help our customers to make better decisions and to be more productive and sustainable. Some of the ways we actually add value to our customers is by improving the way they monitor the farm practices. We can help to improve disease management, optimizing fertilization, because some of our tools are related to the way that the nutrients are working in the soil. Also, we can provide technology for testing ag inputs in general. Could be fertilizers, biostimulants, or even soil disinfectants, any kind of products, natural or any kind of products that our customers are applying can be tested by using our technology, and they can decide whether it's the right product or not for that specific soil. Of course, all of these features help our customers to improve yields and to reach sustainable profitability, which is usually the goal. Regarding the workflow or regarding the process for reaching that goal, everything starts, as I said, with a soil sample. So we receive the sample in the lab, and for us, that sample is information. But why that sample is information? Because we run a process of DNA extraction, so we can collect all the DNA present in that soil. And once we have that DNA, we prepare through different processes that DNA to amplify specific chains that are of our interest. And then we run a sequencing process, which means we read each nucleotide with an equipment from Illumina, and we generate through that process a huge list of DNA chains belonging to different microbes in the soil. And this sequencer is connected to bioinformatics system, and the bioinformatics system will interpret all of this information through these functional predictions. What is a functional prediction? Because this is important to understand. There are different steps in the process. The first step that the system follows is identify the microbes. Just to explain it in an easy way, it is like if the system assign a name and last name to each chain that has been identified from the soil, so that it can build a whole map of the community. It is like a city that with many different citizens, So the system just built all the community and understand who are the citizens in that community, who are the microbes living in the microbiome coming from that soil. Once that is done, next step is understanding the functions of each microbe. We have different and extensive databases with functions for different microbes, so that we can assign a function to each one, and then we can analyze the interaction between each microbe and the community, and the interaction between the community and the crop. This is also relevant because one specific microbe can have some functions in the soil, but those functions will be modified by the environment, by the presence or absence of different or some other microbes in the sample. So this is important to understand. Of course, it's a complex calculus and a complex approach, but once we understand the community structure, the functions of the microbes, and we perform this meta-community analysis, we can come up, or the system actually can come up with this report, which is the BeCrop report, that has three main categories of indexes or information. First of all, we describe the biosustainability of the soil, in which we talk about biodiversity, functionality, and resistance of the soil. Then we describe different health features of that soil. First of all, we list all the pathogens present in that sample and calculate the risk of disease for each pathogen. Because as I said before, the fact that the pathogen is present in the soil is not enough to say that the risk of disease is high or medium or low. We need to understand all the interactions in the community. Other than the pathogens, we analyze the biocontrol effects in the soil, nematicides, insecticides, fungicides, and bactericides effects in the soil, hormone production, which is relevant in the soil, and different indexes related to stress adaptation for the plant. And last but not least, because this is super useful for our customers, we describe all the nutritional pathways for both major and minor compounds. We analyze different features of the microbiome community linked to the each pathway in the cycle of each nutrient. So we can tell in the report whether the soil is supplying the nutrient to the plant or competing somehow with the plant. It could be by blocking the nutrient or by consuming the nutrient through the microbiome. So this is also important to know, because sometimes our customers are maybe applying huge amount of fertilizers, but those nutrients are not reaching the plant, and this is because the soil is not working properly and is not providing or supplying the nutrients to the plant. Other than the report, we offer as part of the BeCrop product, we offer access to an online portal with different online tools that are totally useful for going above and beyond in terms of analyzing the data. There are explorers for look for specific microbes in each sample, co-occurrence tools, heat maps, different tools that are very interesting and enhance a lot the information delivered in the result. Well, now we know what BeCrop is and how it works and the information it delivers. The idea is to describe a little bit a case study that we carried out with our friends in Kairos Vina in Croatia. First of all, some context so we can understand what we are talking about. They took samples from 5 different plots or parcels in 2 different time points. One of the time point was in the summer, in June, and the second time point was in October. There were 3 different varieties in these plots. One of them was Cabernet Sauvignon, the other one was Zinfandel, and then there were 2 plots with Plavac Mali, which is a very popular variety in the Dalmatian coast in Croatia. These are the samples, the date that they were collected, and this is a map of the parcels, and we can see that here they had a slopey terrain, or this is the direction of the slope. So there were 3 plots in the upper position and 2 plots in the lower position. Of course, after receiving the samples, we ran all the analysis, and when the results were ready, together with the technical department in Europe, we analyzed the results, and these are some highlights of the meeting we had with our customer to discuss these results. One of the first approaches that we used is the PCOA, is the principal coordinate analysis, in which we can understand similarities or dissimilarities between different communities of microbes. In this chart, you can see that there are crosses and circles. Circles correspond to samples collected during the summer, and crosses correspond to samples collected in October. And the color of each point tell us the plot or the parcel that sample is coming from. This approach helps us to understand the similarity between the taxonomical profile of each community. So here we can see that in terms of bacterial community, there is a similarity when we consider the samples collected during the summer and the samples collected in October. So we can see that the microbiome is quite different depending on the time of sampling, the time point. Same happens for the fungal community. We can see that for the samples collected in October, the community or the microbial community of each soil, it's pretty similar to each other and completely different from the community of each sample in the summer time point. This is something interesting to analyze. Then, by using one of the tools I mentioned in our online portal, is a heat map. We can look for specific patterns or trends in terms of the different features of the report. In this case, for example, we analyze the biocontrol agents in the soil, and we have this kind of chart in which blue means very high and yellow means very low. So we can see for each column is a sample. So we have the 5 samples from June and the 5 samples from October. So here we can see the differences between these 2 time points in terms of bactericide agents, fungicide agents, insecticide agents, and nematicide agents. In general, we saw that there is an increment in all of the biocontrol agents in October compared to June, and we saw this, which could be sort of alarm or a warning point. We saw that in 2 plots, the nematicide agents in the soil were very high. And although we don't sequence the DNA of nematodes, we have seen, and this is based on our experience with different customers that are using this technology, we have seen that when we detect a high or very high activity of nematicide agents, that could be related to the actual presence of nematodes in the soil. And this is somehow it's because the fact that the nematicide agents are there, it means that they have something to be fed from, So this is, as I said, like a warning. We cannot conclude anything with these 2 samples, but this is something we should pay attention in the future together with the customer. Then, regarding health and more specifically regarding pathogens, we have seen that mainly in June, and this corresponds when the moment in which the plant is more active between these two time points. In June, we have seen a high presence of both ESCA complex and Petri disease. So this is, as I said before, this is the risk of the occurrence of the disease based on different features of related to the pathogen in the soil. This is, of course, a potential for having a disease. We need also the weather conditions, but in case the weather conditions are the right one for these diseases, we can know in advance by doing these kind of trials that the risk of disease is in some cases very high. So this is also relevant. Then, in terms of phytohormone productions and stress tolerance indexes, we have seen that overall in the time point of October, all of these features have grown, are higher, let's say, than in June. And this could be due to many different reasons. One of them is that this June was a very hot June, a bit hotter than the average, and this could be affecting the microbiome and also the moisture in the soil and different features that we should take more samples and analyze in detail. But with this first approach and with this small amount of samples, we can see that there is a clear trend for these features. Then, and this is something I mentioned in the beginning of the presentation, the nutritional pathways. Here we can see that, and this is something interesting because today this is something that it's becoming trendy and relevant in the agriculture industry, which is carbon sequestration, and also because of the profile of Kairos, because they are looking for sustainability and for better farm management practices and stuff. So we saw that in 2 plots in October, the carbon profile, let's say, changed from releasing carbon to sequestering carbon. Of course, as I said, this is a first approach. The amount of samples we took is not so high, but it is interesting because it can provide some hints of what's going on in the soil. Then we saw that the nitrogen, for example, the pathways for the nitrogen were working good in June, and they were turning down a little bit in October. Same happened with zinc, and the opposite happened with sulfur, copper, magnesium, and yeah, that's it, and magnesium. So some of these differences could be related to the fact that in October the plants are preparing for winter, so the activity is quite different. And as you might or might not know, there is a strong communication between the plant and the microbiome through the root exudates. So some of these situations can be explained by that, and some others can be explained by specific characteristics of the soil. I wanted to make the most of this presentation to show you also a feature that we have in our online portal, which is the map, in which we can draw, the plots, the parcels, and we can filter the map by different features analyzed in the soil. In this case, for example, we are seeing the nitrogen. And this map is for the time point corresponding to June. So we can see, as we saw in the heat map, that most of the parcels are working pretty good in terms of nitrogen. They are supplying nitrogen pretty good to the plant. Although there are two that are a bit higher, but in general they are working pretty good. But we saw that for phosphorus and for potassium, these three plots that are located in the upper position in terms of altitude have some kind of problems with the pathways for both phosphorus and potassium, which doesn't happen in these plots in the lower position. I will insist with this because this is important. This is the first approach, and all of these details we are highlighting from this presentation are triggering new questions that we can answer in the future with specific analysis and with more research. But this is interesting because we can see the potential of the technology and the high scope of the analysis and how can we use it for get better insights. Okay, so now this is the summary of what we have seen with Kairos Vineyard in Croatia. And now, as I said before, when we perform this kind of analysis, usually we end up opening new questions and discovering together with the customer new possibilities for the tool and for the technology. So now I would like to present three different possibilities among many more, but three specific possibilities that Kairos could follow right now, based on what we have seen in the first report. The first option, and this would be the most accurate, and it was recommended by our technical department, is to perform a crop monitoring plan, which means selecting specific and relevant time points across the cycle of the plant, of the crop, and collect samples to analyze what's going on in those specific points, so that we could understand the dynamics of, for example, the ESCA complex and Petri disease. Also, we could understand the dynamics in terms of nematicide agents in the soil, because when we do a BeCrop, we get what we get. It's a picture. But when we perform a monitoring plan, we get sort of movie, and we can understand these dynamics of each feature for the crop and for the soil. So this would be a nice approach, and it will also allow us to understand better what's going on with the nutritional pathways. In this case, we could recommend take 4 time points. One could be right now, February, March, when we had the bud break. So the plants start to be active and start to relate with the soil. Then another time point in April. The third in July, which is the time point we collect the samples in the first project, and the last one around harvest could be September, October, and with those 4 time points we could have this better idea of what's going on and help the customer to make better decision in terms of management. Another option, and seeing that there are different soils and so on, could be working to identify the terroir. Our technology allows us to identify the terroir based on the microbiome profile. We have a really extensive database of different soils where vineyards are growing. Soils from Napa in the US, from Spain, Italy, France, Australia, South Africa, South America, many different soils from different regions in the world. And that information allows us to tell our customers whether their soil is completely different to the rest. I mean, it's a unique soil or not. And this is interesting for defining this the terroir of that soil and having a tool for differentiation in terms of commercialization and stuff for the customer. And last but not least, because this is also very interesting, and based also on the profile of our customer and his interest in sustainability and better agricultural practices and so on, we could go and try to understand the profile of each plot in terms of its management. And by doing this and by running this kind of analysis, we can determine in which, let's say, how close is a specific plot to one of these three kind of management: conventional, organic, and biodynamic. And we do this by analyzing the structure of the community and the profile of that microbiome. I will briefly explain this. In this case, for example, we can see these three clusters. Each point in this chart represents a microbe, and the color of the point represents the function of the microbe, and the lines represents the connections between one microbe and the community. So here we can see that under a conventional management, there are some clusters. The biodiversity is not so high, and the functionality is also pretty low. And this is because of the selection pressure that the agricultural practices have been putting on this microbiome. So this is very fragile. If we somehow modify some of these clusters, the balance of this community will be broken very easy, and this means that this soil is more suitable for the establishment of pathogens, is less stable in terms of functionality and so on. When we analyze an organic soil, a soil under an organic management, we can see that we still see some sort of clusters, but now there are much more dots, much more points, much more functions, and the interconnection between each microbe and the community is much higher. So this structure is stronger, and it's going to be harder for example, for a pathogen to establish in this sort of community. And of course, when we take a look at the soil under a biodynamic management, we can see that there is no clusters, it's almost like one big dot. So this is a rich soil, a very stable soil with a high biodiversity, with a high functionality. And of course, this kind of soils will require less inputs than this soil. So by running this kind of analysis, you can know, or a customer can know, where each plot is located, or closer to which one is located, and make decisions. Well, this is pretty much it so far. I will be happy to take questions and clarify anything it's needed. And well, hopefully you have enjoyed the presentation. Thank you very much.
Thank you, Nico. Really interesting. There are a couple of questions. As we are in the interest of time, I think let's go for the part of Ljubo. It will give more context and more information about this specific case, and then we will jump into the questions, whether you can answer it or maybe Ljubo can also answer them. So I'm gonna put Ljubo here in the stage, and also close. You can close your sharing screen if you want.
Yeah, it's
okay,
here.
Great. Yeah. And then I call Ljubo to the stage. Thank you, Nico, and welcome, Ljubo. How are you?
Hello, do you hear me?
Yes.
Yes.
All right. Hello, everyone. My name is Ljubomir Jelavić, or shortly Ljubo Jelavić. And I'm here on behalf of a company I run, and it's my family's company, one enterprise which is situated on the central Adriatic coast of Croatia. And in the next 10 minutes or so, I'm going to give you a roll-up of who we are, how did we came into the soil being the pillar of hopefully being a pillar of all our viticulture activities. Then what insights did we gain from this particular collaboration with Biome Makers? And at the very end, something about what the future pathway could be, at least from our point of view. So, all right, when we talk about who we are, actually, it pretty much boils down to the question of where we are. So, our geographical location and the history of it gives much of a clue in that regard. So we are part of a split municipal area, which is the second largest urban area in Croatia. And traditionally, historically, that was that was an area of very pronounced Mediterranean agriculture activity, dotted with small but very small urban settlements, and all that came to halt after the Second World War, after which this wave of modernization, which actually meant a lot of industry coming in, basically it changed the culture of the whole area, and it just meant a sort of a rift in the community, which isn't healed until this day. It's a topic in the community. And long story short, when my family and some of their friends contemplating about planting a vineyard, they started from this position, the position of someone trying to restart something which was basically a mainstream couple of decades ago, and which was no longer a case. And the year was 2007 and 2008, and they happily underwent this plantation of the vineyard. They found a beautiful spot on the slopes of one of the hills that surrounds this coast around the city of Split. But they made a mistake. They made a couple of mistakes, which I'm among them, who came after them are trying to erase. Just what they made as a mistake in the first place. They actually started as a conventional agriculture production, and they went into it as a default position. They didn't see it as a problematic thing to do, which obviously was, and I will tell you a little bit about it later on. Another problem they brought us was the very spatial layout of our vineyard, which was basically put right on the slope, without actually thinking that might cause a lot of problem with erosion. And you might ask yourselves how they didn't envision it. But I have to mind you that there were people involved from various aspects of scientific community, who gave us their reassurances that won't be much of a problem. But fast forward to say 2016 to 2017, we've lost a lot of topsoil due to erosion. So we've lost a lot of humus, and this is one of our problems we need to solve as soon as we can. So just allow me just one step back. When I took the reign of a company in 2014, one of my first decisions was to transition ourselves to organic agriculture, because I saw it as a huge potential, and I knew we could do it. And but this is not enough. So basically, the problem we face is how to redefine our viticultural activities beyond organic, but based on data and scientific proven scientific activities, right? And so enter the microbiome assessment, right? That's something we've been eyeing for some time. And as soon as we figured out that there is a Fields Forever tender out, we've applied, and we were granted two analysis of our soil samples, right? And Nico told you a lot about it. Now, what did it bring us? It brought us a lot of insights, and some of these insights are direct, some are less so. And when I say direct, I mean the insights on top of which we can start a real action, right? Obviously, those so-called direct insights are in those realms of health pathways and disease. Those less so are those that come under the term of biosustainability. But that's because we didn't think about biosustainability before, and even those data, those insights about biosustainability will be very important for us, because on top of those insights we will build at least new knowledge and nomenclature, and we'll start to think about it. But the clearest path is in the realms of health and nutrients. And if we talk about nutrients, we've gained very interesting insights in terms of what happens to our carbon, nitrate, potassium, and phosphorus, and those later two are of crucial importance for us, because there is an amount of it, to say it, in our soil, but we can't reach them at the moment. But at least now we know what kind of microbiome sits in our soil, and we know are they beneficial or not in terms of trying to use them to get those potassium and phosphorus elements in our soil. But the real breakthrough came in terms of health, and it came in terms of something that we thought we were helpless about, and this is ESCA disease, which hit us. ESCA disease being actually a very known disease, vineyard disease, but until very recently, it was the disease that hit very old vineyards, and probably due to climate change, it starts to hit younger and even young vineyards as well. And the problem is the science still doesn't have much of a clue. And with this insight that came from microbiome assessment, we've managed to pinpoint the worst hit areas of our vineyard, and to gain insight how to analyze the rest of the vineyard that wasn't sampled, the areas that weren't sampled, and based on the insights from those sampled areas, we figured out which were actually which wines, and I'm talking about the single wines, which wines were actually hit by ESCA complex. And as we speak, we are undergoing an activity of getting rid of those wines which are under the disease, right? So, long story short, as Nico told you before, we opened much of newer questions than we gain answers. But nevertheless, we know in which direction to further analyze, and that's a huge step forward. And as I've said, we even gained a particular insight which gave us a direct actionable data. That one that's in regards to ESCA complex. So at the very end, just please allow me to share with you my thoughts about how to scale this a little bit up. I talk a lot with my colleagues in the area, winemakers, wine growers, olive growers as well. I told them everything I've learned about from this project, and we all came to the similar conclusion. And the conclusion is, in order to scale this, we think both users and the providers like Biome Makers would benefit from public, so state or EU level help in terms of market pull and push. And at least in the EU, there might be something interesting in that regards on the horizon, and it has to do with how EU define a new innovation policy for the next 7 years. And this is so-called mission-driven innovation policies. They have five missions on top of which they build their innovation policy. One of those missions is called soil, right? And when you dive into the position papers and regulations, and the regulations already started pouring out from Commission, you see one of the pillars is soil monitoring. And one of the pillars of soil monitoring is microbiome assessment. And what they envision is a network of so-called living labs, basically a sort of innovation platforms in local areas, and they envision about a dozen per particular one member state of the EU, which will organize from bottom up on farm, landscape, and urban realm soil monitoring, right? And this is something we will definitely be interested in exploring further, and actually would like to talk with our partners for Biome Makers about diving a little bit more into that and scaling this very interesting path for agricultural activities further.
Thank you, Ljubo. Yeah, very interesting thoughts.
Thank you very much.
And very interesting also to know that we are opening more questions for you to work on that and being able to work in a sustainable way in your vineyard for now and for the future. So thank you. We are just a couple of minutes before we close this webinar. So let's go into the question round. I'll call Nico once more to the stage. And so I will go for questions for Nico or either you, Ljubo. Let me just go and put you in half. Yeah, there you go. Okay, so let's dedicate another 5 minutes. It's almost an hour have passed since the beginning, but I think it's worth it to dedicate a couple of more minutes to some of the interesting questions that we have.
Of course.
Great. So let's go with the first one, asked by Vittoria Bardelloni. That she said, I think, like at the middle of your presentation, Nico, related to this heat map that you were showing. She's asking, what is the best moment to sample soil if I want to see if the mycorrhiza products I applied in my vineyards are affecting the microbiome or not?
Okay, well, there are two different approaches in this question. The easy answer is that we should test the soil right after applying the mycorrhiza product. I mean, right after could be 15 days after the application or 20 days, depending on the product you are applying. Because as you have seen in the presentation, the microbiome is really dynamic, and if we can analyze the soil before and after applying the product, we can see specifically the difference that the product has provoked in the soil. That said, it's important to know that sometimes there are some products, like mainly biological products, so-called probiotic products that could be mycorrhiza or some other fungi that could last in the soil depending on the inoculation process. So sometimes we cannot find the mycorrhiza itself in the soil after a few months, for example, but we can check the effect of that product in the soil in terms of the function. And this is important to clarify, because as everyone have seen in the presentation, we analyze not each microbe isolated. We analyze the community and the function in the soil. So the fact that a given product could be a mycorrhiza, modify that microbiome can tell us that, for example, some specific features that could be phosphorus mobilization or nitrogen mobilization, or even some feature related to hormone production, can be modified. And that change in the soil can last differently depending on the product and in the soil conditions. So it's also interesting to see the effect, and it's also interesting to measure right after applying the product, and also later in time, to see whether it's necessary maybe to re-inoculate, the soil.
Okay, thank you,
Hopefully
Nico.
it's clear.
We have another two questions done by Krishan that might be similar to what you just said. So I think it's worth it to compile them both and ask it to you. So Krishan is asking, do we get quantitative data for different mineral elements? And also, how we get mineral elements, taking into account that what we are doing is a DNA sequencing?
Well, maybe it wasn't 100% clear in my presentation. What we are analyzing through the analysis of the DNA is the pathways for the minerals in the soil. We are not analyzing the actual amount of nitrogen, phosphorus, or whatever in the soil. What we are telling to our customers is, okay, if you have enough amount of nitrogen, for example, in the soil, your pathways are working for the plant or are working against the plant. We always recommend to combine this microbiological analysis with a physicochemical analysis, which will tell us specifically the amount of nutrients that you get in the soil. And then the way we relate the microbiome community with with how the nutritional pathways are working is through different portions of the DNA that we can identify, and we can, of course, based on different scientific references and trials and stuff, we can identify these specific genes that are codifying for specific enzymes that are related to each pathway in the cycle of the nutrient. Let's say in the cycle, the easiest is the cycle of the nitrogen, and we say we have a nitrification, ammonification, different pathways, and each pathways will be executed by specific enzymes, and those specific enzymes will be released by specific microbes through, or the fact that those microbes will release those enzymes is determined in the DNA of those microbes. So by analyzing that, we have actually 4.5 million of genetic references, and we use those references not only for identifying microbes. We identify about 100,000 microbes, but also for identifying specific functions, mainly related to nutritional pathways. So this is the way we relate the DNA with the minerals.
Christian is also asking, and then what is the cost associated with this analysis? What would be the pricing if he wants or she wants to do one single analysis?
Well, this is very, very transparent. The price per sampling for BeCrop is 199 euros. Each sample must be a composite sample, so you must take a few sub-samples, at least 4. We recommend taking from 8 to 12. Yes, that's the price. Usually we define the projects, not one single sample, but yes.
Yeah, and this is maybe for Ljubo, because we have another question related to sampling, which is how is this process and how should you do sampling in order to get results? So maybe you can answer that part.
It was very easy. It's straightforward thing to do. We received a kit by post, and it came with instructions. It instructed us how deep we should dig, and everything was very, very clear and straightforward. And we just packed the samples and sent them back to Spain, to Valladolid. And in very short time, we had our data, and so if I'm allowed, just one more thing to add, we've gained a lot of insight due to very, very good portal and the way how it's designed. So it's I really recommend this to anyone interested, and it might help you a lot.
Thank you. I will add something to what you said, because it's true that it's really easy to. The sampling process is quite simple and easy. Actually, the depth from where you have to take the samples is 15 centimeters at maximum, like from 10 to 15. So it's quite dynamic and it's very easy to collect. Then depending on specific projects, we can take samples from the rhizosphera or specific parts of the soil, but in general we take just 15 centimeters. There is a very descriptive sampling guide, so it's quite easy.
Okay, thank you. We've passed 7 minutes from the hour that we were finishing, so I just want to thank you once more to be here, to all of the audience. I hope you have now a clear view and you gain some insights for you to improve your management practices or your agricultural practices in general. The questions that couldn't be solved, please, you can send us those questions through our web form in biomakers.com. And if not, we would love to see you in our next webinar in a couple of weeks. So thank you once more, Ljubo and Nico, and have a nice day.
Thank you very much.
Thank you. Greetings.